here at the University of Nairobi as a BSc student, finishing with a bachelor's in science in biochemistry, and then went on to Oxford and worked on malaria immunology. I think that's what you did, Valentina, and then got the Gates Cambridge Scholarship and other scholarships to study at the University of Cambridge. And Valentina has now submitted her thesis, so I think it's out for examination. But she has offered to present what she has been doing for the last 4 years. So it's really a pleasure to have you, Valentina, joining us today. Part of the team that's attending the meeting here are people looking to use applied epidemiological modeling skills and to learn epidemiology and use it in their own countries. We have, I think, colleagues from Rwanda, from Ethiopia, and a few other places. So it's nice to have you here. You might inspire a few more people to do like what you have done. So we'll give you some time to give your presentation, and then we shall have an amount of time that we spend talking about any questions that these colleagues might have. So please go ahead. And hi, Andrew. It's good to see you.
Awesome. Thank you so much. This, I'll just go ahead and share my screen. Just let me know when you can see my PowerPoint. And there's some videos here, so just let me stop sharing and share again to allow the audio. Okay, so you should be able to see my screen now. That's fine. Okay.
Yes, you can see. Yeah.
Right.
Perfect.
So hi everyone. So today I'm going to give you a brief overview of my PhD work and share with you a little story about a recent public engagement project that I did in Northern Uganda. And the topic of my PhD is modeling the spatial and temporal distribution of Bacillus anthracis suitability across Uganda and Kenya using a Bayesian approach. So I'll spend the first 40 minutes or so taking you through the work that I've been doing for the past 4 years and use the remaining 5 minutes to talk about a public engagement project I recently implemented in Uganda. Anthrax is a zoonotic disease caused by Bacillus anthracis, a gram-positive, soil-borne and spore-forming bacteria. And the image above just shows the life cycle of the bacteria. This soil-borne pathogen can inhabit the soil for several years or even decades under ideal conditions, greatly increasing its risk and making long-term control of the disease very difficult. The disease primarily affects both domestic and wild animals, particularly grazers, and the animals likely become infected when they ingest spores from contaminated soils, vegetation contaminated with emesis from necrophilic blowflies, or via bites of hemophagic flies. So when the spores detect favorable conditions within the infected host, they undergo germination to produce a vegetative form that multiplies rapidly, a process which can lead to the death of a susceptible host. And upon death, the carcass of the host sheds the vegetative bacilli, which sporulate into resistant spores, causing environmental recontamination. Now, anthrax outbreaks are usually associated with a small number of species, even though the bacteria affects a vast range of animals. And the underlying reasons are poorly understood, but could be linked to different levels of exposure and susceptibility amongst hosts and differences in circulating strains. So anthrax mostly infects ruminants and ungulates such as sheep, cattle, goats, pigs, horses, deer, hippopotami, zebra, buffaloes, warthogs. So anthrax usually manifests in animals as sudden death without prior clinical signs, and the carcass usually appears bloated with bloody and clotting discharge from body openings and no rigor mortis. Other signs include mouth and throat ulcers, and this is particularly in pigs and carnivores. Now, humans display 4 forms of the disease, as previously mentioned. 4 forms of the disease. So cutaneous, gastrointestinal, inhalational, and the rarest form, which is injection anthrax, observed in heroin injection drug users. The picture on the top here shows a local who participated in the slaughter of an animal that had died of anthrax, and he later developed lesions in his hands that formed painless ulcers with black centers. And the picture on the bottom here shows an example of such an ulcer. So anthrax is responsible for about 20,000 to 100,000 cases annually across the world. And about 63.8 million poor livestock keepers and 1.1 billion livestock live in anthrax risk areas globally. Now, anthrax is both preventable, it can be controlled, but surveillance systems for the disease are still poor. And although geographic extent of anthrax risk in Kenya and Uganda has been studied previously, past methods have been unable to capture the underlying spatial and temporal dependencies within the ecological data sets because the previous algorithms do not have the capacity to do some of the rigorous methods required to detect these dependencies. So ecological niche modeling using INLA, integrated nested place approximation, a spatially and temporally explicit approach adjusts for these structural dependencies, producing more precise estimates to facilitate targeted surveillance and prevention. So given this problem, my overall aim is to accelerate anthrax prevention and control efforts in Uganda and Kenya by designing nationwide risk maps and investigating predictors of anthrax occurrence and incidence using ecological niche modeling and applying a Bayesian approach. So the outline of my PhD work is as follows. I have a systematic literature review followed by 4 main result chapters. And in the systematic review, I look at all the research articles that have been published starting from 1980 to 2021 on anthrax niche modeling. And here I assess the reproducibility, the quality of input data, the algorithms applied, model assessment, and output approaches selected. And in my first result chapter, I apply a conventional algorithm called maximum entropy, Maxent, to create a model to predict the distribution of areas suitable for the occurrence of Bacillus anthracis across Uganda. In the second result chapter, I apply now the spatially explicit modeling framework using INLA to design a similar model across Uganda, but this time adjusting for spatial autocorrelation, a non-bias that affects the performance of models. In the third chapter, I apply INLA again, but this time in Kenya, to create a spatial model of Bacillus anthracis suitability, and then apply this model to predict areas across Kenya suitable for the occurrence of the bacteria. And within the same chapter, I also discussed the results of a spatial temporal model of anthrax risk using population-based socioeconomic predictors of anthrax risk at sub-county level. In my final chapter, I discussed the results of a knowledge, attitudes, and practices survey that I conducted in Northern Uganda to understand the factors hindering effective anthrax prevention and control in the region. Now, for the sake of brevity and interest, I will limit this presentation to the first 3 modeling chapters and the systematic review. So I will start with the systematic review. And here I did a literature review of all research articles focused on niche modeling studies of Bacillus anthracis. And the review was limited to the peer-reviewed papers published from 1st of January 1980 to December 2021. And I included only articles published in the English language or those with English translations, and selected only research-focused articles based on correlative niche modeling, which summed up to 28 papers. Then I narrowed down the following review questions. The first one was, which scientific journals are selected for the publication of niche modeling of Bacillus anthracis, and when have this been published? And secondly, how reproducible are the research articles? Thirdly, what input data, that is species occurrence and environmental variables, have been used to study the topic? And fourth, what algorithms have been applied for model calibration? Finally, which model assessment and output methods have been applied? So the journals that are most frequently used were PLOS, Neglected Tropical Diseases, publishing 6 out of the 28 papers, then PLOS 1, Preventive Veterinary Medicine, and the American Journal of Tropical Medicine and Hygiene. The figure on the right shows the number of articles published against the year of publication, and the overall number of peer-reviewed publications has gradually increased from just 1 paper in 2007, published in American Journal of Tropical Medicine, to 5 articles in 2016, and 6 articles in 2020 across a variety of journals, and only 3 were published in 2021. And I also measured reproducibility across the selected articles by identifying and recording the checklist items reported in each paper, following the recommendation by Feng et al., who published a study on the recommended items that should be reported in niche modeling studies to maximize reproducibility. Now, the y-axis here shows the various reproducibility checklist items to be compared, and the x-axis shows the number of articles that reported those items. So these are labeled from A1 to D5, and A1 is at the very bottom. So A1 to A72 are those related to the species occurrence data. Then a B1 to B4 are related to environmental variables used. C1 to C7 describe the algorithms and model calibration. And finally, D1 to D5 describe the items related to model assessment and output. So for occurrence data, A1 to A72, out of the 10 recommended checklist items, spatial autocorrelation and download date of occurrence data were not reported across all papers. All the 28 articles, however, reported the basis of the records and the spatial extent of occurrence data. 27 studies mentioned the source of occurrence data, and 26 reported the temporal range and sampling bias. 18 articles reported spatial uncertainty, and 19 used methods that removed duplicate coordinates. Only 3 articles reported in depth how they dealt with spatial outliers. For environmental variables data, all studies reported the source of environmental data, and 25 reported the spatial resolution. 20 studies mentioned temporal range, and only 2 articles reported the download date or version of the data source. Out of the 7 checklist items for algorithm and model calibration section, all the studies reported the algorithm name and provided a justification for variable selection. 27 studies mentioned the extent of background data, 11 reported the number of background data used, and 16 reported the sampling method for background data. 18 articles reported the version of algorithms used, and 25 the parameterization. So finally, for the 5 checklist items under model assessment and output, the evaluation index, the dataset used for evaluation, and the output format was reported across all studies. However, only 16 articles used a threshold-dependent evaluation index, and only 8 reported a binary threshold output. Now, next here is another barcode that shows the percentage of checklist items reported in each individual study. So you can view it as sort of a reproducibility score, and it provides an illustration of the performance of each study in terms of reproducibility. And you can see the individual scores reported varied between 50% and 88.5% of all checklist items. And then the most common environmental variables used across studies were temperature and precipitation, soil properties, vegetation, and topography. Only 4 studies used hydrology variables, and 7 used biotic variables such as host availability, meaning livestock and wildlife densities, and human impact. And the pie chart on the left shows the different types of algorithms that have been used in the past for model calibration. The most common algorithm choice was Maxent and GAP. As for the model assessment methods, the bar plot on the right shows that AUC was the most selected metric for the evaluation of niche models across studies, followed by omission and commission, which were used in studies that applied GAP algorithm. So my overall aim was to apply a relatively new approach using INLA, which has more robust capabilities as compared to the conventional methods. But first, I designed a niche model for Uganda using Maxent. And then in order to compare the results with another subsequent model using the INLA approach. So in my first chapter, I looked at the geographical extent of Bacillus anthracis suitability in Uganda using Maxent. The map above shows the distribution of anthrax case locations across Uganda. Navy blue circles show the wildlife cases, 294 of them, used for model calibration. And the green triangles show the livestock cases, and yellow diamonds here represent human cases. The blue polygons show the locations of the 50 kilometer, 75 kilometer, and 100 kilometer background buffers, which were constructed around the wildlife cases that were used to calibrate the model. On the human and livestock data were recorded 8 years after the wildlife data. This is because data collection in Uganda before 2018 was not very systematic. So human and livestock data were recorded 8 years after wildlife data and were therefore temporally distinct from the wildlife data. And these were withheld from model calibration and used to evaluate the final model. So out of all the 3 environmental predictors that I use, Mean AVI and distance to water bodies contributed the most towards predicting anthrax suitability. And elevation had a smaller contribution towards the final model prediction. The bacteria tends to exist closer to water bodies at an elevation of between 42 to 253 meters above sea level, and a mean AVI of negative 174 to 5,400. So according to this map, most parts of central and eastern Uganda could be suitable for the occurrence of the bacteria, as well as large pockets in the north and southwest of the country. So just to reiterate, we used data from wildlife outbreaks that occurred between 2004 to 2010 to calibrate the model. And this model was able to predict livestock and human case locations occurring 8 years later. This means that had this model been developed in 2010, then the catastrophic 2018 outbreaks that occurred in Uganda might have just been anticipated and prevented. Then I estimated livestock populations at risk by overlaying the maps of livestock numbers with a binary map of anthrax suitability using a threshold that maximizes sensitivity and specificity. So across Uganda, the results showed that anthrax risk areas contain about 1.5 million livestock at risk. And these include about 600,000 cattle, 670,000 goats, just about 112,000 sheep, and over 180,000 pigs. So that concluded the Maxent study. I then ran a similar model, but using INLA this time. So the second chapter, I looked at the spatial distribution and predictors of Bacillus anthracis stability across Uganda, but now using INLA. I specified the number of background points and randomly selected them from the target buffer ready. I referred to these as pseudo-absences, and the ratio of presences to pseudo-absences was 1:1. And the navy blue circles here show wildlife cases used for model training, and the blue triangles show livestock cases, red diamonds here, human cases. So this is just the same data set. The blue polygons show the locations of 50 kilometer, 75 kilometer, and 100 kilometer background buffers that were constructed around the wildlife cases, leaving 10 kilometers between the buffer and the presence locations. So the pink dots show the pseudo-absence points within the 50 kilometer buffer, orange dots within 75 kilometers, and the white dots are the pseudo-absences within 100 kilometer buffer. So I used almost similar environmental variables to the Maxent model. The only difference here is that I use soil calcium and not EVI, because these two are highly correlated. So using them interchangeably really does give quite a similar result. And also in terms of prediction, the model preferred soil calcium. So I also added temperature seasonality, but ended up dropping it in the final model because it was not contributing much to the model's performance. I then applied a Bernoulli model with both linear and non-linear effects for the covariates and a spatial random term as shown in the equation. And the figure on the right shows a mesh structure used to estimate the spatial random field. The posterior mean and 95% credible intervals were estimated for all the parameters. And the best model used the 75 kilometer target buffer radius and elevation as a non-linear effect, and distance to water, soil calcium, and soil water as linear effect. So option 8 in the table. So eventually, the spatial random effect was not included in the model because it didn't really add much to the model fit after the addition of the covariates. The posterior predicted mean probability of Bacillus anthracis stability and the 95% credible intervals for the parameters of the fixed effects using the final INLA model are shown above, and the results demonstrate a negative relationship between distance to water and the presence of Bacillus anthracis between 0 to 0.3 kilometers. However, soil calcium had a positive association with the occurrence of the bacteria between 10 to 25 units. Similarly, the results showed that higher occurrences of bacteria are expected in areas with soil water availability of between 20 to 30 volume percentage and elevation of 140 to 190 meters. The figures above show the posterior predictive mean and the posterior standard deviation of the probability of Bacillus anthracis suitability across Uganda. The AUC value for the best model was 0.9, indicating very good model performance in terms of excellent discrimination ability between presence and absence locations. And the sensitivity of the final model against withheld evaluation dataset was 90%. As for the prediction, you can see that it's almost similar to the Maxent model output, aside from the fact that its prediction is more generalized and it picks up more known locations of past anthrax outbreaks as compared to the Maxent model. Now, this is a binary version of the same model, and the red areas here are locations predicted to be suitable for anthrax. As you can see, down at the bottom in the south of Uganda, the model is able to pick up areas of suitability in Kiruhura and Rakai districts. So I mentioned this because these are known hotspots and cases have been recorded here. But during my 2019 fieldwork trip, I wasn't able to retrieve the records from these two particular districts. However, the model was able to pick them up quite successfully. Now, one key limitation of this study is the lack of systematically collected and detailed anthrax case data. Therefore, it was impossible to account for other structural dependencies, such as temporal autocorrelation. So in the next chapter, I applied a more detailed and systematically collected data set of anthrax occurrences in Kenya from 2006 to 2020 to create a spatial and spatial temporal model of anthrax incidence and occurrence using INLA. And the granularity of the data set also allows us to demonstrate how spatial temporal structures can be applied to improve the model fit of ecological niche models. So I analyzed the long-term spatial data set, a beautiful data set of livestock anthrax cases collected over 15 years in Kenya. And I built two models. So first, a spatial model to explore associations between anthrax incidents across Kenya and ecological covariates, then a spatial temporal hurdle model, so comprising two models, an occurrence and incidence model to investigate the socioeconomic drivers of anthrax occurrence and incidence at sub-county level. So analyze records occurring from 1st January 2006 to 31st December 2020 across Kenya, collected from the Directorate of Veterinary Services in Nairobi and the 5 regional investigation laboratories located in Nakuru, Eldoret, Karatina, Kericho, and Mariakani. I extracted data from old paper records of anthrax cases into Microsoft Excel. So some of these records were really old and hard to retrieve, but we got them. So originally we had data from 1990 to 2020, but we chose to use, to subset this to use only the data from 2006 to 2020. So I recorded the geographic coordinates of the cases at district level and removed duplicate coordinates, outliers, and entries with missing variables. In the end, I had 540 livestock cases that I used for analysis. I also collected wildlife data from Kenya Wildlife Service, and I recorded 20 wildlife case locations that I used to validate the performance of the spatial model. So start with the spatial model. I created a 50-kilometer buffer polygon around the observed livestock cases, then created a 20-kilometer by 20-kilometer square grid structure within this buffer and counted the number of points within each grid cell to obtain a given number of counts per cell, and then extracted the coordinates of the centroid of each cell to create marked locations with a given number of cases per location. So I essentially converted the data into a count process. So this is number of outbreaks per location. And in the end, I had 95 cells with one or more counts, which formed the new presence locations. And then randomly selected 95 pseudo absences within the 50-kilometer buffer polygon, but at a distance of 10 kilometers from the presence locations, as shown in the map with the white triangles. And then modeled anthrax occurrence as a function of EVI, elevation and distance to water. I defined a zero inflected Poisson regression model with spatially correlated random effects, implemented as a generalized additive model, GAM model, with the anthrax incidence as the response variable. So the model is defined as shown in the equations, where CI here is denoting the observed number of cases at location I. So to test whether the addition of the GAM smoothers and the spatially correlated random effects improved the fit of the model, I also considered candidate models without smoothers and spatial random effects as shown. So I tested 3 versions of the model. The first used distance to water elevation and EVI as linear covariates without spatial random effects. So basically a generalized linear model. And the second applied nonlinear terms to elevation and EVI without spatial random effects. The final one was similar to the second model, but now with the addition of spatial random effects. I then measured the DIC values of the candidate models to select the final spatial model. So basically what I expected to see is the DIC should have been getting smaller if the fit was improving with the addition of these aspects. The final spatial model with the lowest DIC had distance to water as a linear effect and EVI and elevation as nonlinear terms with the addition of spatially correlated random effects. Now the posterior means and the 95% credible intervals for the fixed effects parameters used in the final model are shown in the table on the bottom left. And the estimated model demonstrates a negative association between distance to permanent water bodies and anthrax incidence. However, EVI had a strong positive association with anthrax incidence. Similarly, increasing elevation was also associated with an increased incidence of anthrax disease. So I used the final model to predict anthrax incidence across Kenya. And the figure shows the posterior mean and the uncertainty around the slope of the posterior mean incidence of anthrax disease across Kenya. So I'll explain this more. So the gray areas here show locations where greater than 5% in A and greater than 10% B of the slope of the predicted incidence was below the cutoff for positivity, and this was 0.2. So I extracted the fitted density distributions of the predicted anthrax incidence, so the slope, and calculated the proportion of the density distribution that was lower than the positivity cutoff, and then grayed out areas that had more than 5% or 10% of the fitted density below the cutoff. So basically, in other words, saying these areas had more than a 5% or more than a 10% chance of having zeros. So the scale for anthrax incidence shows colors ranging from blue to red, with blue areas showing those areas with low incidence and warmer colors towards red, showing areas with higher incidence. And the specially projected nationwide risk maps from the best model identified large parts of Western Kenya, Rift Valley, central and coastal regions to be at high risk of anthrax disease. And the INLA model was able to identify locations of wildlife outbreaks held from the model calibration, such as Shompole Wilderness, Garissa, Tsavo National Park, Meru National Reserve, Kielu Kaluku, Hell's Gates, Lake Nakuru National Park, Nairobi National Park, Mbagathi and Srisambu Conservancy, and some of the counties with the highest predicted anthrax intensity were Narok, Bomet, Kisii, Kericho, Nandi, Uasin Gishu, Kakamega, and Kisumu, and the southern regions of West Pokot and central Trans Nzoia and Busia counties, which lie next to the eastern border of Uganda, were also predicted to be at high risk of anthrax. Now, several counties in central Kenya were also identified as high-risk areas, and the model also picked up unknown anthrax risk areas across parts of Samburu, Turkana, and Marsabit counties. This is towards the north of the country, so arid and semi-arid lands. Also, most parts of coastal Kenya were identified as high-risk areas, including Kwale, Kilifi, Mombasa, and Lamu. Also, the southern regions of Garissa County were also identified to be at risk of anthrax occurrence. Okay, so then my second objective was to investigate the socioeconomic population-based drivers of anthrax risk at sub-county level, and this is part of the spatial temporal model. Therefore, I developed a second aerial model with the number of observations per sub-county as the new response variable. So I analyzed the whole monthly case time series from 2006 to 2020 and mapped out annual counts of confirmed and suspected anthrax cases across Kenya at sub-county level to analyze the spatial and temporal trend throughout the surveillance period. Due to the sparsity of data, I aggregated monthly case counts and modeled the quarterly occurrence and incidence of anthrax at sub-county level scale, including spatial and temporal effects, to determine the socioeconomic drivers of anthrax disease risk across Kenya. So overall temporal trends suggest that 2006 to 2010 appear to be distinctly different from previous years, with markedly high peaks in confirmed anthrax cases. Now, throughout the study, 86 sub-counties in 26 or 47 counties reported confirmed anthrax cases, with evidence of marked spatial as well as temporal clustering. However, 204 sub-counties reported no confirmed anthrax cases. Most cases, 65%, were reported from only 25 sub-counties within 13 of the 47 Kenyan counties. And these 13 counties were Bomet, Kericho, Kiambu, Kilifi, Kirinyaga, Laikipia, Mombasa, Murang'a, Nairobi, Nakuru, Nyeri, Tharaka Nithi, and Uasin Gishu, with much lower incidence in the northern counties. So I employed a two-stage modeling approach using the Hadl model distribution to separately model anthrax occurrence, presence or absence, across all sub-counties via logistic regression, and incidence counts using a zero-inflated Poisson distribution. I modeled the quarterly anthrax incidence. So basically had 17,400 observations. This is 290 sub-counties over 60 quarters. So I modeled this quarterly anthrax occurrence using a Bernoulli distribution as shown in the first set of equations, and then modeled the quarterly anthrax incidence counts using a zero-inflated Poisson process with parameters as shown. I modeled the Bernoulli and zip distribution separately as functions of the covariates and the spatial and temporal random effects using a general linear predictor. So here alpha just denotes the intercept, X is a matrix of socioeconomic covariates accompanied with the linear coefficients denoted as beta, and spatial temporal reporting trends at the sub-county level were accounted for in the model using spatially structured and unstructured random effects specified jointly as Bessak-Yolk-Molly BYM model, as well as temporally structured random effects, the Y term, of the first order, where W here is just a pure noise term that is normally distributed with a mean of 0, variance of sigma squared. And I used an informative priors with Gaussian distributions of the fixed effects and penalized complexity priors for the hyperparameters of the random effects. And the models with the socioeconomic variables explained significantly more of the variation within the data set compared to the baseline intercept only model. So even here I had candidate models. So the model with the covariates improved the DIC by 338 for the occurrence model and 332 for the incidence model. Then the addition of the spatial random effects and the temporal trend further improved the model fit compared to the one with the covariates alone. And this improvement was by 583 and 591 for the occurrence and the incidence model in terms of DIC. So the occurrence and incidence model with the covariates, spatial random effects, and the temporal trends were selected as the final models. So to examine the robustness of these models, I then looked at the sensitivity of model outputs to geographically based cross-validation by fitting separate models in turn, holding out those high burden sub-counties from each of the 11 sub-counties that had high reported anthrax incidents. The table here is showing those 11 counties and the associated sub-counties. These are Nairobi, Kiambu, Murang'a, Nyeri, Tharaka Nithi, Nakuru, Kericho, Uasin Gishu, Bomet, Kilifi, and Mombasa. So I withheld this data one at a time and checked again the fixed effects, magnitude, and direction. And what I expected to say is, if the model was not influenced by one geographical location, these fixed effects should remain the same. And this was the case. So the magnitude and direction were robust, across all holdout models, showing that the findings were not overly influenced by data from any one geographical location. So the figure shows the spatial distribution and correlates of annual anthrax occurrence and incidents at sub-county level across Kenya. So maps show the fitted probability of anthrax occurrence and incidents for the 290 sub-counties in the last quarter of 2020. And the points and error bars in C illustrate the posterior mean and the parameter estimates of the fixed effects and the 95% credible intervals for the best fitting models of anthrax occurrence in red and anthrax incidents in blue. And the linear covariates were standardized, centered and scaled prior to model fitting such that the parameters estimate the effect of one standard deviation change in the covariate on either the odds of anthrax occurrence or on anthrax incidents. The models both included spatial temporal random effects. This is sub-county per year to incorporate the spatial and temporal heterogeneity. And the findings from the spatial temporal occurrence and incidence models show that anthrax risk is strongly influenced by an increasing total human population, which may increase demand for meat and hence the host population sizes, animal-human contact, and access to healthcare. Similarly, there's a substantial positive association with the number of exotic dairy cattle, which can also influence host population sizes and human contact. The negative association between anthrax risk and the population density could be linked to increased risk in rural areas, which are less densely packed and have reduced access to healthcare and anthrax awareness programs, and also have more livestock in general compared to urban areas. And the negative effect of increasing number of livestock producing households on anthrax risk could be due to increased awareness of anthrax in areas with greater numbers of livestock farmers. Or similarly, this might also explain the negative association between agricultural land area and anthrax occurrence. So although the socioeconomic covariates used in the models can explain a bit of the pattern of anthrax occurrence and incidence, they do not fully account for the observed spatial distribution of anthrax, and there's still some residual spatial variation that is not explained by those fitted covariates. Consequently, the addition of the spatial random effects and also the random walk trend further improves the fit of the model. The figure on the right here is showing that random walk trend, and it also shows that there was a downward trend in anthrax cases from 2006 to 2012, and including this information in the model produces a much better model fit. So basically, in summary, the burden of anthrax disease is global, and Uganda has an estimated 1.5 million cattle at risk, but no policy on routine vaccination of livestock. Kenya has reported yearly cases of anthrax since before 2006, with less than 10% of the sub-counties accounting for about 65% of cases. So our INLA provides almost similar results to the Maxent model, with added ability to investigate the presence of spatial and temporal autocorrelation within data sets. Addition of spatial and temporal random effects can improve the fit of niche models, and also socioeconomic risk factors should be considered when developing interventions for anthrax prevention and control. So in terms of the strengths and limitations of the study, so detailed systematic literature review of Bacillus anthracis that includes information on reproducibility hadn't been done before. So this provides a first attempt of that. And also, it also gives a first presentation of the model of the national suitability of Bacillus anthracis across Uganda using our INLA. And then also here apply a large and systematically collected data set spanning 15 years to develop the first spatial temporal model of anthrax incidents and occurrence at sub-county level in Kenya using INLA, as well as a spatial model using the same approach to predict nationwide geographical incidence of anthrax across the country. So some of the limitations were the lack of systematically collected surveillance data for Uganda. So basically, I ended up removing the spatial random effects because there was no evidence of further spatial autocorrelation after adding covariates. But there's a possibility that had the data been systematically collected, maybe we would have been able to see some spatial autocorrelation and adjust for it. Then another limitation is the assumption that the regional laboratory confirmation of anthrax is uniformly representative across all sub-counties in Kenya. So these findings have relevance to 3 key policy questions. And number 1 is, should policymakers rely on the results of past models and maps of anthrax risk? Okay. For Uganda, the models demonstrate that the nationwide predictions generated by Maxent and INLA models are quite similar. This is partly because the same dataset was applied, almost similar variables were used. The spatial random effect was eventually excluded in the INLA model. But it is important again to consider that lack of systematically collected surveillance data in Uganda could have impacted the ability to detect structural dependencies within the data. And with more detailed data, it could be possible to make more precise estimates of anthrax risk in the future. It's also important to note that the Kenyan INLA model identified areas that have also been previously identified by past studies, which is very good. So the bottom line is that our INLA is not a replacement for the conventional modeling frameworks, but it is a novel tool that should be used alongside classical algorithms like Maxent, boosted regression trees, and GAP because of its robust capabilities. The second question is, should policymakers in Kenya improve surveillance of anthrax in marginalized areas of the country, such as Turkana and Samburu? Well, the results from the Kenyan study demonstrate with a high degree of certainty that northern parts of the country, specifically across Turkana, Samburu, and Marsabit counties, were at high risk of anthrax incidents. These areas comprise arid and semi-arid land with mostly pastoralists who are often economically and politically marginalized, lacking access to both veterinary and public health services that are available to the rest of the country. So these pastoralists are at a greater risk of both the risk of zoonotic diseases such as anthrax, and the sparsity of recorded anthrax cases in this region could reflect limited surveillance practices and not necessarily the absence of anthrax cases. So basically, the absence of evidence is not the evidence of absence. A key recommendation from this work would be to improve anthrax surveillance across all of the areas identified in the model. Then the last question is, should public health programs aimed at reducing anthrax pay more attention to socio-economic factors such as human and livestock population sizes and agricultural activities? Well, the results seen from the Kenya study suggest that anthrax risk is strongly influenced by increasing total human population and the number of exotic dairy cattle. The results also suggest a negative association between anthrax risk and population density, increasing number of livestock producing households, and agricultural land area. Therefore, public health initiatives aimed at reducing human-animal contact, improving access to healthcare, and increasing anthrax awareness may have a positive effect in terms of reducing anthrax occurrence and incidence. And basically, this brings me to the last part of my presentation, which is the public engagement project. So I applied for and received some funding from the public engagement team here at Cambridge and traveled to Uganda to conduct a 2-day event comprising a sensitization workshop to create awareness on anthrax, followed by a cattle vaccination program the following day. So this was inspired by the survey, which I've not presented here from my last chapter, which identified a serious knowledge gap and also poor practices and attitudes of farmers within this area. So I decided to do this project. And my team and I conducted baseline and follow-up surveys to evaluate the effectiveness of the workshop, and preliminary results showed that the workshop was very effective in improving the knowledge of farmers with regards to anthrax transmission, effects, prevention, and control. The following day after the workshop, we traveled to remote villages where farmers live to conduct the first ever farmer-led vaccination campaign in that region. So it was farmer-led because we let the farmers form groups and set the agenda for the program. On our side, we offered logistical support and subsidized the cost of the vaccine by paying 50% of the cost of vaccination for each animal. So to put this into perspective, it costs $1 to vaccinate each animal in Uganda. So he paid 50 cents on the dollar for each and subsidized the cost for the first 300 cows, summing up to about 100 pounds, and the turnout superseded our expectations. So we coordinated with the area vet. Hope this video will play. We coordinated with the area vet and 2 animal health workers. At some point, they let me vaccinate one of the cows. So being from a non-vet background, this was very wild and interesting. Here is a short video of that.
No, you first have to prick. Hard.
And then this next video is showing one of the villages that we visited. Again, these areas are very remote, hot, and far from the central districts, about 2 hours away from the nearest town, Arua. So this has heavy implications on routine vaccination programs because a vaccine like anthrax requires a cold chain storage. And then this final video was some feedback we got from one of the farmers.
The program is good, but today we know how to inject animals.
Anything else? How many of your animals were vaccinated?
Me, mines are 10. Yeah, they are 10, but we are requesting you to vaccinate all the villages in our parish.
Okay.
Yeah.
And would you recommend for other farmers to also do the same for
Yeah.
their animals?
Yeah,
Why?
but the others get information now, but they complain about money. But my request is we bring program again.
And what do you think about the price of the vaccine? Is it high for the farmers?
No, it is good. They agree. 1,500 is okay.
Okay.
But the others are never get the money today.
Okay.
And with that, I would like to acknowledge the following. My supervisors, James and Andrew, and Dave Redding from UCL, as well as the team at Makerere University, the CDC Bacteria and Special Pathogens Branch, the Gates Cambridge Trust, and the Royal Geographical Society for funding this study. Thank you very much.